Our next speaker, the last one for the morning, is Antoni Ivorra. He's a full professor here at the Department of the Information and Communication Technology. He's like home of the University of Pompeu Fabra. He completed his PhD in electronics engineering from the University of Politècnica di Catalunya in 2005. And after that, he enjoyed 4 years of postdoc at the University of California at Berkeley. And also he followed up by an 8 months postdoc position in France at the CNRS Institut Gustave Roussy. And from there, he came back home here in Spain, in Barcelona, and he joined the University again of Pompeu Fabra, where he rose and grew from a researcher to the full position professor that he has now. And during his research years, he has been an inventor and co-inventor of 12 families of patent application. And also in 2016, he was awarded an ERC Consolidator grant for his research on microstimulator for neuroprosthetics. His research is focused on bioelectrical phenomena, and in particular on exploring the use of these phenomena for implementing new methods and devices for biomedical application. His main topics are electroporation for tissue ablation, electrical bioimpedance for diagnosis purpose, and injectable electronics for neuroprosthetics. And today, he will present some of the modeling methods he's using to predict electroporation in living tissues and how these methods have been used to create new therapeutic approach and also for personalized treatment planning. The floor is yours.
Thank you. Thank you very much. Let's wait for the presentation. It's over there, I promise. But please. So just to start with a kind of a disclaimer, I'm not an expert in modeling. So basically in our group, we do a number of things. Basically modeling for us is just an aspect, so don't expect much, sorry for that, in terms of modeling. But I will present what we do in the field of electroporation in terms of modeling. So yeah, let's wait for a while until we get the computer going on. So you will need probably the presentation again. So just let me allow just to start a little bit without the slides, the introduction. So my name is Antoni Ivorra, it has been presented. So basically in our group, the Biomedical Electronics Research Group, what we do is basically to study bioelectric phenomena, and particularly the applications for these bioelectric phenomena. And we now have two main research, let's say, topics. One of them is electroporation, which is what I'm going to talk today about. So you're going to learn about what electroporation is. And the other topic that we do is injectable electronic implants, which is what is related to the ERC consolidator grant that was mentioned. I'm just going to put just on a slide. We also do modeling in that area, but the truth is that most of our modeling is into the electroporation side. So that's why I decided to both, let's say, this talk to electroporation and to see what there and what not. I see it. So it's starting. Yes, very good. So now we are going to say it here. You're going to see the title. So it's an honor to be here. Thank you very much for the invitation. It does not happen frequently that have the opportunity to give a talk here at home. And we have kidnapped some students, I see. That's good. Good. So that's the title. I'm going to talk about modeling basically of electroporation-based treatments. And basically it's going to be a talk, an introduction to electroporation. So this is the snapshot of the website of our small group, like 8 to 10 people. And as I said, what we do is bioelectric phenomena, study bioelectric phenomena. And currently we are basically focused on electroporation and injectable electronic implants. This is just the only slide I'm going to show you about these electronic implants. What we are doing is, as I said, injectable implants, so implants that can be deployed by injection because they are very thin. And we achieve this level of minimal invasiveness with these implants because basically we are substituting the methods that are used until now to power implants. In order to power implants, you either have a battery or have a wireless power transfer method, which until now has been basically inductive coupling. So you generate a magnetic field, you pick up the magnetic field inside the implant, or ultrasound transmission. In any case, all those methods at the end need a bulky part, a bulky component inside the implant to pick up the energy. So basically we are doing something completely different, which is to apply high frequency currents of high magnitude across the body. And these currents are applied in short episodes, short bursts, and because they are high frequency and they are applied in short bursts, they are innocuous. The patient or the subject doesn't even feel them. It's at the implants where those currents are transformed into low frequency currents that are able, for instance, to perform a stimulation. And that's what we mostly are interested. We are interested in having implants that act as microstimulators for neuroprosthetics. So what you see on the right, sorry, here, it's one of our latest prototypes, which has been implanted in many animals. We also have tested that technology in humans, and now basically we are moving into exploring applications. So if any of you have any idea of applications for microstimulators, let me know. Good. So I switched to electroporation, which is going to be the topic here. And well, I guess some of you are familiar, but electroporation is a phenomenon. Basically, electroporation, it's the increase in cell membrane permeability in a non-selective way to ions and molecules when we apply an electric field, a high electric field. This, typically the electric field we apply very short in time. Typically we apply this electric field in the form of pulses. So basically we have our cells in a cuvette, or we have the cells in a tissue. We put there 2 electrodes, and we apply a voltage or a current so that we generate an electric field in tissue or in the cell suspension. And depending on the magnitude, we can get what's called reversible electroporation on the top, which basically implies that there is a transient period in which the cell membrane is permeable, but after the transient period, the membrane reseals and the cell is perfectly viable. And this is used for different applications, like gene transfection, in vitro and in vivo gene transfection. And then we have irreversible electroporation, which is basically killing the cells, means killing the cells. We kill the cells directly, let's say through a necrotic process, or we kill the cells through a regulated cell death mechanism. That can be apoptosis, but it seems that other mechanisms are also involved there in what you see. And this is interesting, and this is an area in which we in particular are working. This field of non-thermal irreversible electroporation as an ablation mechanism. So basically, the talk here is going to talk about electrochemotherapy. I'm just going to describe in the next slides what electrochemotherapy is, and irreversible electroporation as an ablation mechanism, as a mechanism to destroy tissues. And we will see that it's basically meant for tumor eradication and for cardiac ablation. So electrochemotherapy, it's a technique that basically started to be developed in the 90s, and it's now quite common to be found in hospitals in Europe for treatment of surface tumors, basically skin tumors and head and neck tumors. And basically, Lluís Mir, a Catalan guy that for a long time has been in Paris, developed that, or pioneered the development of this technique there. And the idea is to use reversible electroporation. So the idea is that we inject a chemotherapeutic, in this case bleomycin, which does not penetrate well into the cell, normally, systemically around the body in a low dosage. And then At local level, we apply, where the tumor is, with electrodes, we apply the electric field so that we promote the uptake of this drug, and there we kill the cells that are growing a lot, basically, the cells that are proliferating. And as I said, this is a technique that is used with very, very good results. The other thing that we are going to talk about, it's about irreversible electroporation. Irreversible electroporation came a little bit later, and at the beginning, it was known that if we applied high electric fields, we would kill the cells directly. But for many years, no application in the field of medical devices was seen for it. Basically, it was considered something not wanted to achieve. But then Boris Rubisky at Berkeley, with some other collaborators, including a PhD student, Rafael Davalos, they understood that maybe irreversible electroporation was interesting as an ablation modality. There were, at the time, and there are still, many different physical ablation modalities, like cryosurgery or radiofrequency ablation, which are based on thermal changes. Either you freeze or you warm up the tissues, you heat the tissues to destroy the tissues. So those techniques work, but they have an inconvenience. The inconvenience they have is that they not only kill the cells, but they also kill or destroy the extracellular matrix up to a point. And this is different for the case of irreversible electroporation, because in irreversible electroporation, what we are killing is the cells directly. So the extracellular matrix is preserved. At the same time, if there is any kind of heat sink phenomena close to blood vessels, this is not going to impact on electroporation, which is something that happens with cryosurgery. And this first advantage of not having an impact on the extracellular matrix is very important, and it explains to a large extent its success. And here you see an example of a very early study in which I participated, in which what we did was to treat the carotid artery of a rat completely with a very intense electroporation. Later I'm going to talk about the intensity of electroporation. And what you see is, in the normal rat, you expect to see smooth muscle cells where the wall of the artery is. And with irreversible electroporation, we basically remove all those cells. Those cells are not present. You don't see the nuclei anymore. But the structure of the vessel is still there. It's still patent, and it's working as a vessel. So the rat survived for weeks without any issue. And this demonstrates how powerful it is in that sense. So irreversible electroporation now is used for tumor destruction, okay, for solid tumors. In particular, it's very commonly used in prostate tumors, basically because it does not destroy the nerves, and that's important compared to other techniques. This is now, there is only, or until recently, there was only one company that held all the patents about irreversible electroporation as a method to kill basically tumors or soft tissues. And well, it has been developed, but I have to say that it has not been developed as much as it was promised in the past. So there are people is treated all around the world, but the truth is that it has not exploded as it looked in the past. The typical procedure is what you see here with multiple needles that act as electrodes. You go to the tumor or the periphery of the tumor, and you apply with a generator that you see here on the left these high voltages in the form of a sequence of pulses. These pulses typically are what you see there in the order of 100 microseconds at our frequency rate or the repetition rate of 1 hertz, more or less. And you apply like 80 or 90 of those pulses. And this destroys the tumors. It has been a complete success, and this is a more recent story, is what is called a PFA, post-field ablation, which is another name that they are using in the field of cardiology for irreversible electroporation. So when irreversible electroporation was proposed a few years ago, in 2004, as a method to destroy soft tissue, of course, there was an interest in looking into whether it could be used for cardiac ablation. But the results were not convincing until a company came, which was Farah Pulse, and they started to demonstrate that the technique was very useful, and now all the major companies, medical device companies, are behind that. Now, so there are a lot of startups and a lot of major companies now with activities in the field of post-field ablation as an ablation modality for cardiac tissue to manage arrhythmia. So typically, you go, as you see here, the typical procedure that you want to perform is what is called pulmonary vein isolation, where you have foci for generating arrhythmias. So you want to isolate those veins, electrically, let's say, isolate those veins. So you want to kill the cells. but you don't want to damage, for instance, the esophagus. And this is what it's achieved with PFA. With PFA, you kill the cells, let's say, in the pulmonary veins, but you don't destroy the esophagus, which would be very problematic, in some cases fatal. This is booming. As I said, I mean, I was like 2 weeks ago in a major conference on New Orleans in cardiology, in heart rhythm management, and this was booming. It's surprising to see this field so, and also quite appealing for a biomedical engineer. So I'm now going to the main topic, which is modeling, okay, of electroporation. So there are different types of modeling that we can perform, or that people, let's say, it's performing. One important type of modeling is to understand the electroporation phenomenon. We still don't understand what's going on. And there was even discussion for many years whether this poration, meaning that we are creating pores, was real or not, okay? Now the agreement is that, yes, there are pores. And to a large extent, it comes from molecular dynamic simulations, which I'm not going to talk, I'm just going to describe. So in these molecular dynamic simulations, it's molecule, it's atom of water or ions. It's modeled with classical physics. And these molecular dynamic simulations, what they show is that when we apply a voltage across the cell membrane, so what you have here is the cell membrane, when you apply a voltage very rapidly, high enough voltage, of course, very rapidly small channels of water appears, and those channels expand and create what are called hydrophilic pores, which were, let's say, forecasted many years before. So these hypotheses that were done years ago were confirmed by molecular dynamic simulations. What the molecular dynamic simulations don't show, and are still not able to reproduce, is the fact, so they reproduce the fact that electroporation is very fast. Once we have the transmembrane voltage reaching a transmembrane voltage of around 200 millivolts, we know that there is electroporation happening very fast in a matter of fractions of nanoseconds. And that's what molecular dynamic simulations tell us. What we also know from experiments is that after removing the electric field, there is still some status of permeability. So the electroporation, so this state of increased permeability can last for seconds, minutes after application of the pulses. And the simulations don't show. The simulations, the molecular dynamic simulations, indicate that after removing the electric field, the poration, let's say, vanishes completely. So that means that something else is happening here, and what it's now thought to happen is that there is some chemical transformation of the lipids. In particular, it's thought that there is a peroxidation of the lipid tails because of the electric field and the presence of water inside the membrane. Good. So one thing that we can model very easily, and even with analytical models in some cases, is what is the transmembrane voltage that we have when we apply an external electric field? Because I've said electroporation starts to happen when the transmembrane voltage reaches a threshold. And this we can model, and we even have analytical models. And there is a very nice equation, very simple, that indicates for the assumption that the cell basically is an extracellular medium which is conductive and intracellular medium which is conductive. So we are talking in both cases of ionic solutions. And then we have a thin membrane which is a dielectric. So assuming that we have a sphere with conductive media inside, conductive media outside, and a thin dielectric, we have this expression for the transmembrane voltage. And this expression for the transmembrane voltage is telling us that in the poles of the cell facing the electrodes, we have maximum transmembrane voltage, and in the poles against the electrodes, we are not going to have any transmembrane voltage, right? Because it's going to be zero. And this is what we get. Experimentally, it has been observed, looking with dyes that mark or indicate the transmembrane voltage, we see that that's a reality. So that's very interesting. The thing here, or one aspect to take into account, is that this transmembrane voltage is not reached immediately. When we apply the electric field, this structure of this extracellular medium, the intracellular medium, and the dielectric is a capacitor, so it takes a while to charge this capacitor. So typically, this charging time is going to be in the order of microseconds or fractions of microseconds, so that you have an idea. So this, I mean, there's a community of people doing simulations on that. We apply an electric field, we have different cell shapes, and we see when electroporation occurs or not. And there is also a community of people working on doing very basic simulations of the electroporation phenomena. Okay? This is not what we are interested in. We are more interested in studying in our case, in our particular case, electroporation, although we have performed some simulations of cell level, we're more interested in studying what happens in tissues, okay, because it has more practical. I have some water, please. So basically what we can say is that we are interested, let's say, in the macroscopic electric field. So when we have a tissue, like on the top, and we apply an electric field with 2 needles, we assume that the tissue is homogeneous. We are going to have an electric field distribution as the one that you see here on the bottom left. So this, inside the tissue is heterogeneous, we have the cells, it's going to create the non-uniform fields. But if we look at the transmembrane voltage, we are going to find the same that we had for these isolated cells. So for this isolated cell, what we had was that the transmembrane voltage is proportional to the electric field. And here, when we have a tissue, it happens the same. So when we have a tissue, the transmembrane voltage that we have in the cells in different points is proportional to the local magnitude of the electric field. So at the end, that indicates, and experimentally has been demonstrated, that we have electroporation where the electric field goes beyond the threshold. And that's the basic assumption that we do when we do simulations. So our basic assumptions to predict electroporation in a tissue is that electroporation is going to occur in a specific point of a tissue if the electric field goes beyond the threshold. Of course, that depends on the kind of tissue, the kind of cells, and also depends on the protocol that we apply. So typically, we don't apply a single pulse, but we apply a sequence of pulses, because that has been demonstrated to be more effective to achieve electroporation. And at the same time, so once we have a sequence of pulses, we have different features. We have the different size of the pulses, the shape of the pulses, the frequency. So for a specific protocol, we achieve electroporation if the electric field goes beyond the threshold. This is a typical plot that we have typically in which we demonstrate that the electroporation phenomenon is dependent on the electric field magnitude and the exposure duration. But even for a single duration, we can play a little bit around there with the different waveforms and exposure characteristics. So for low electric fields, we don't do anything. For higher electric fields, we get reversible electroporation. For higher electric fields, we get into irreversible electroporation. And for much higher electric fields, we are going, or very long electric fields, we are going to get into thermal damage, which is something that we always want to avoid. In any case, for electroporation therapies, either if we want to do gene transfection or we want to do irreversible electroporation, we are going to have 2 thresholds, a maximum that we don't want to go beyond and a minimum which we want to be above. Right? We want to be sure that we cause irreversible electroporation, but we don't want to cause thermal damage. And this is what typically we do. It's as simple as that. So how we do that? Typically, the community uses is a finite element method. Here, everybody is very familiar. What we use is what sometimes is called the Laplace equations, what you see here on the bottom. And typically, the truth is that we use commercial packages or made packages like COMSOL or Elmer to solve this problem. So we set up our geometry of our electrodes, we set the properties of the materials, and we apply voltages or currents, and we see what's the electric field distribution that occurs, and where the electric field is above a threshold, we have electroporation. Where the electric field is beyond, we don't have electroporation. So I'm going to talk a little bit about how these models have evolved. Of course, since the beginning, it was understood the necessity of playing with heterogeneous materials. So it was understood that different materials have different conductivities. So that was incorporated since the very beginning. But even if we assume that the tissue is homogeneous, which is useful for many cases, in many cases, we just want to study or perform numerical studies with homogeneous tissues just to study the geometry of the electrodes that we want to apply, for instance, and to see if we are going to cause thermal issues. So at the beginning, we assumed that the conductivity of tissues was constant, okay? But that's not true, because once we have electroporation, there is an increase in the permeability of the cell membrane, and there's going to be an increase in the permeability of the cell membrane to ions, okay? So at the end, we are going to have quite a large increase in the conductivity of the, so we are creating, let's say, in other words, a short circuit in the membrane. So this is important, and we and we started, let's say, to work on models that incorporated this change in conductivity during the delivery of the pulses. Because as I said, electroporation is very fast. So once we apply the pulse, at the beginning, we already have electroporation. We already have an increase of conductivity. So that was, what's going to be the consequence? The consequence is that we apply an electric field. This electric field changes the conductivity, and this in turn has an impact on the electric field distribution. So I'm just going to show a very old, because that was proposed. Many of us started to work on those models. But the truth is that we didn't have evidence that that was really important. So we conducted many years ago a study, very simple study, to demonstrate that that was important, that this change in conductivity. And for that, in a lot of cases, we use animals. In this case, we decided that it was not worth to use animals, so we went to potatoes. Potato, it's an interesting model for us because it exhibits electroporation. So the cells, so you take a piece of meat, you are not going to see electroporation there because the cells are completely destroyed after a few hours. Okay, they burst, basically. They are bursted. But the plant cells survive. Basically, you cut them, and the cells are perfectly viable for many days, months. So that's the case of potato. So we can perform electroporation in a potato and kill the cells in a potato. And what happens when we kill the cells in a potato is that after a while, they become dark. So we can visualize very easily where we have electroporation. So what we did in this model was to compare the conventional method up to that point, which was to assume that the conductivity was constant with time, with the new refined method, which now is not so new, which was to include this change in conductivity due to the electric field. For doing that, the first thing that we did was to study how they become dark, the potato becomes dark, when we apply a completely uniform electric field. We can force a uniform electric field, which is applying to plates. So we have two plates, like the setup there. We know that doesn't matter if the conductivity changes, the electric field is uniform. And from that, the samples become dark, and we can, let's say, do a metric for this level of darkness. And the other thing that we did was to do recordings while we were applying the currents for electroporation. So we saw how the conductivity increases because of electroporation. Okay? So now we have a model, and what you see here on the right is what we typically see. We see shape like it's like a sigmoid, in which for low electric field, there is no change in conductivity, and then the conductivity increases with the electric field magnitude in a very step way, and then it saturates. Once we have, let's say, porated the cell membrane, doesn't matter if you create more pores, this is not going to add more conductivity to the tissue. Let's say the cell membrane is too short-circuited. So we incorporated that, and we compared, and we did a comparison, which we have, I don't know how visible it is here with the light, but what you see on the left are actual pictures after treatment, so like 24 hours after treatment, we take the pictures, and what you see here on the right are simulations. In the middle, you have the constant conductivity model, and on the right, you have the model that includes this change of conductivity due to electroporation, and you see that there is a very significant improvement. And now this is something that we always basically use when we want to do accurate simulations of electroporations. I'm just going to introduce here an example of use of the modeling for electroporation, which is also serves as an example for patients, and you will see why I say that. So when we do electrochemotherapy, we have to, as I said, a lot of times is done for superficial tumors, head and neck tumors, and some cases we use needle electrodes that create like 2 rows of needles, but sometimes we use plates, okay? And if we can squeeze the tumor between these 2 plates, and so that it creates like a kind of regular geometry here on the left, the electric field distribution inside the tumor is very uniform, and then we have a very successful treatment of the tumor. On the other hand, there are tumors that are hard or are very irregular, and when we put in between those 2 plates, there are spots in the tumor that are not treated. And if you have an area that is not treated, you leave some cells not treated, you know that you are going to have a tumor again. Actually, there are pictures in which there is a single treatment, and after the treatment, you have 2 tumors. And so very dramatic illustrations of that. So many years ago, I came with the idea of using, in general, gels with modulated conductivity to kind of distribute the electric field. So because that's what we want to do. We want basically to have an electric field in the tissue that we want to treat, and then to minimize the electric field outside of this tissue, the other tissues. So for that, I said, okay, let's play with gels with different conductivities. And for this specific problem, I said, okay, let's fill, if we have an irregular shape because we cannot squeeze the tumor, let's fill all the dead spaces with a conductive gel, which has a conductivity very similar to that of living tissues. A very simple idea. With that, we would have a very regular electric field distribution, very uniform, so that we would treat the tumors very effectively. The thing is was, okay, before going into, say, experimentation, up to which point this conductivity has to be equal to the conductivity of tissues. And now I said the conductivity of tissues increases with the electric field. So which one I have to pick up? The other question is how tolerant the technique is to misplacements in the gel. What happens if I apply too much gel, if I apply too little gel, or the gel is not creating like a rectangular shape? So this is something that simulations allow us to do. And this is basically the results of simulations. In gray, basically, what you have is the target electric field. So there are two ranges. So there is a minimum threshold, maximum threshold. So what we want is to have our tumor in gray. And what we saw is that, well, okay, the technique was quite tolerant to conductivity mismatches. Right? If we apply a conductivity of the gel that is kind of close to the conductive tissue, we are going to have a very substantial improvement. And then we also simulated the case of different depositions of the gel, and we also saw that it was quite tolerant to irregularities in the deposition of the gel once it was sufficiently covered. And with that, we decided to go to an experimental data, and it worked basically with a very suboptimum treatment for electrochemotherapy. So remember this technique in which we inject bleomycin and then we apply the electric field. We saw that by applying the gel, we improved a lot the efficiency of the method. And I was super happy because I said, okay, gels were already used to improve the contact between the tissue and the electrodes. I was in a place that people was working with clinicians in. I said, okay, well, this technique is going to be adopted immediately and we are going to benefit patients. But no, it took many, many years. And for a few years ago, I started to realize that people was using this technique for veterinary patients. Electrochemotherapy, it's very commonly used in veterinary patients, dogs, cows, horses, because their radiotherapy, it's not an option in many cases because of the size, the cost. So because radiotherapy is not an option, in a lot of cases, electrochemotherapy is used in veterinary patients. And that technique of using gel was applied there. But I was not aware of any treatments in humans until recently, so like 15 years, after 15 years. So the technique is now in use. This is from a colleague in Argentina that they are using this technique. So as I said, an example of patients in biomedical engineering. So the models now are even further, let's say, refined. Now we also not only include the increase in conductivity of the tissue due to the electric field, which is the thing in the middle, but we also include the conductivity increase due to temperature increase. So when we apply an electric current to a living tissue, to any conductor, we are going to warm up that conductor. We are going to apply heat. So it's going to increase its temperature, particularly where the electric field it's very high. So the heating is proportional to the square of the electric field magnitude. So basically around the needles, we are going to have a very high heating. So there, we are also going to have a conductivity increase. Ionic conductors, like living tissues, when you increase the temperature, they increase their conductivity. So now we are also including that thing into the models. And for instance, this kind of modeling is included in something that we did here at UPF with Miguel Angel and with a student that I'm now going to mention, that it's a treatment planning tool, complete treatment planning tool for electroporation therapies, in which you can segment the tissues, apply the different properties, and then place the needles for applying the treatment, and simulate the electric field distribution and to see if the tumor is covered or not. This is kind of the work, basically, of Enrique Pereira, and this is basically the workflow. It's very, very classical in that sense. And before that, this is a desktop tool, let's say, that we can use in our computers. Before that, Enrique created a much simpler tool, which is online available online, which you can even play with it. And in this tool, basically, we cannot do segmentation. This tool assumes that all tissues are homogeneous. The images that you see here are just an overlay, just to see the electric field distribution. So we can place the needles, we can put a tissue that changes its conductivity during the electric field, but we assume that everything is homogeneous. In that sense, it's very simple, but it's very, I think, educational in particular for clinicians. So last, I have time? How much time do you have? Yes, I have time. Okay, I'm going to explain a last thing. Now I'm going to talk about the disadvantage of electroporation and how we solve this disadvantage. So the thing here is that we have two phenomena. One side, on the left, we have electroporation, which is a universal phenomenon that happens when the transmembrane voltage goes beyond the threshold. And there on the right, what we have is another phenomenon, which is electrical stimulation. So electrical stimulation, it's the triggering of action potentials because we apply a current. So in most of the cases, this is intentional. We want, for instance, in our microstimulators, we want to create action potentials in nerves to create contractions of the muscles. So, but in all of those cases, this is not wanted. And this is the case here. So electrical stimulation is not universal. It only happens in excitable tissues, in nerves and muscle cells and myocytes. So both of them basically happen when the transmembrane voltage goes beyond the threshold. Okay, in electroporation, we say that typically when we go beyond 200 millivolts, we start triggering electroporation. It's not really a sharp threshold phenomenon, but kind of. And in the case of electrical stimulation, it happens the same. When we depolarize the cell membrane, which is to change the transmembrane voltage, we trigger the action potential. What happens is that the threshold for electroporation is higher than the threshold for electrical stimulation. So very typically, when we want to do to apply electroporation, we are going to trigger action potentials. So this is what they say here. Typically, when we apply monophasic pulses, like the one that you see here, we are going to stimulate peripheral nerves, and this is going to cause pain, this is going to cause muscle contractions. Let's hope this works. So what you see here is a human subject, so this is a treatment in kidney, I think. And what you see here is this nanoknife, they called treatment. Let me put it again. And you see that it contracts, and the patient, he contracts. So the patient is contracting, and this is after muscle relaxant. So here, the patient was under strong muscle relaxants. If not, the patient would jump, literally, because we have done that in other animal models, and we see that the animals jump, basically. And this is kind of avoided with muscle relaxants, but you see some movement, so the needles can displace. And to have muscle relaxants limits a lot the applicability of electroporation. And that's one of the reasons why at least this method has not evolved so much. The other method was because the company didn't make the effort to simplify the life for clinicians. To put these 4 needles in a perfect parallel way, this is tricky for clinicians, so they don't want to do that. So fortunately, some years ago, by the same guy that was doing during his PhD proposed to use irreversible electroporation as an ablation technique, had an idea, which for a lot of us didn't make any sense at the beginning, which was to use a high-frequency biphasic pulse. He called that H-fire. And the idea here is that he started doing experiments in which he was applying sequences like that. And what he observed was that not only he was able to cause electroporation, so he was able to treat tumors, and at the same time, he was minimizing a lot, and in some cases, completely avoiding contractions. By the way, we also did some work on that, and we observed that the same phenomena can be observed with other biphasic waveforms, for instance, sinusoidal waveforms. This is we are working on cardiac electroporation. This is a work by Tomás García, in which you are going to see the comparison between a monophasic treatment, so conventional, let's say, pulses, with biphasic pulses, and you will see that there is a very significant change in the contractions. So this, you see the animal jumping. As I said, in the case of a pig, it jumps even more, okay? This is without muscle relaxants. And in the other case, you see that there is movement, but it's minor compared to the other one. So the technique works, and at the beginning, for a lot of us, the question is, how is it possible? I mean, that's why we thought, no, that's not going to work, because if at the end we want to reach a transmembrane voltage, so if we want to reach a transmembrane voltage, we have to apply an electric field. If we apply high-frequency pulses, we know that we are going to minimize the stimulation, because that's a known fact. This is what we use in our microstimulators. We apply high-frequency biphasic AC waveforms, and we don't have any stimulation. So that's a known fact. But at the same time, we said, okay, yes, you are going to minimize the stimulation, but you are not going to produce any electroporation. But the thing is that, yes, electroporation occurs while we minimize stimulation. And this is something that we want to study a little bit. And for that, we decided to start with a modeling study. And I asked Pooja, a very productive PhD student here, to do a study on that. And basically what he did, it was to make a model in which we had both electroporation and electrical stimulation. So basically, he applied different kind of waveforms and created a model for a nerve and for nerve terminations. You're going to see how we did that. So basically, for computing the reversible electroporation or the treatment, we use what I described before. We compute the electric field distribution with finite element model, and we see where the electric field is beyond a threshold for a specific protocol. And these thresholds, we took them from experiments. There were experiments published. This is a work that actually we did with the group by Rafael Dablos, who is the guy that had this idea of this H-fire in Virginia Tech. And what we did, it was then to include what's called the cable model for modeling an axon. Basically, an axon or a nerve, so a continuous axon or a nerve termination. I cannot go through the details, but this cable model basically has the model for the generation of the action potential and the propagation of the action potential. So we basically create an electric field. We put the voltages that we obtain in our homogeneous model, we put them into the nodes of this model, and then the model tells us if there is an action potential and how it travels. We can see how the action potential travels. This is very nice. And what we saw is that, as expected, if you apply biphasic waveforms, so here you see that we are studying the case of two nerves and at two locations compared to the two needles that we are using for treatment. What we see is that as we reduce or we increase the frequency of the pulses, we increase a lot the threshold that we need for stimulation. And this is what we expected. And this is published, and this is what we expect. And in particular, there is a huge difference between monophasic pulses and biphasic pulses. At the same time, what we saw is, okay, we said, let's see what is, if we assume that we have these two nerves at 1 centimeter, let's play with different protocols, and let's see, by increasing the voltage for each one of those protocols, how much I can treat before I trigger an action potential. How large is the ablation volume around the needles I can treat until I reach stimulation. And what we saw is that, as expected, according to results of Davalos, when we reduce the pulse length, when we increase the frequency, we can treat a larger and larger volume of tissue. Okay? So that means that we can we can increase the volume that we can treat just by minimizing the pulse length or increasing the frequency and not inducing an action potential. It's more illustrative the case of nerve terminations. In nerve terminations, what we can plot is, okay, we have an area that we want to electroporate. I said I want to have always for different protocols. For instance, this is the monophasic protocol here on the right, which is the conventional protocol. I want to have this small area in red of the treatment. So I want to see if a nerve termination around this area would be activated. And the model expects or predicts that a large area around the IRE area, it's going to trigger stimulation of those nerve terminations. So meaning that if we have this red area, anything that is within the black circle is going to be activated. Any nerve termination that is there is going to be activated. What we saw with these high-frequency protocols is that when we increase the frequency, the area in which we are going to cause a stimulation, it's reduced. So another demonstration that by increasing the frequency, we reduce a lot the chance of stimulation. So at the end, this is related basically to the fact that with the frequency, we see that the threshold for electrical stimulation increases very abruptly, while the threshold for IRE, for treating the cells, does not increase so abruptly. And the question is why? So what we believe now, or we are pretty sure, is that what happens here is that when we want to treat the tumor cells, basically we are talking about round cells, okay, which are relatively small, while the axons are very long cells. And in long cells, large cells are in principle, if the pulses are very long, long cells are easier to electroporate because the transmembrane voltage, if you see here on the expression, it's proportional to the radius of the cell or the size of the cell. So if we have a long cell or a large cell, in principle it's going to be easier to electroporate because the transmembrane voltage that we are going to induce for the same electric field is going to be larger. But on the other hand, you see that it takes a while to charge. And this is the difference. The thing is that large cells, like the black here, are going to reach, in theory, a larger transmembrane voltage. So they should be easier to electroporate for a long pulse. But if we observe what happens when we are talking about short pulses, we see that the small cells charge to a level more rapidly than the black cells, which is the large cells. So here what it's indicating is that by applying very rapidly changing polarity fields, what we do is we charge and discharge the long cells without consequences, while the small cells are subjected to electroporation. And with that, basically, I'm concluding. If any of you is interested in electroporation, there is a society, there is a conference, there is a very nice school in Ljubljana. And of course, I'm open to any questions. Thank you very much.
Okay, thank you for this interesting talk. Does anyone has a question? Okay, I will start. It might be a very naive one because it's not my field of expertise. Does it work for every kind of cells? Like, could we treat hypothetically cancer?
No, we are treating cancer.
Okay,
Electroporation is already being used to treat solid tumors. As I said, it's used for prostate, it's used for liver tumors. Irreversible electroporation is used in particular for prostate, liver tumors, pancreas. There is a lot of hope with pancreas. Brain tumors, the results are not so convincing. In the case of electrochemotherapy, which is the combination of reversible electroporation with bleomycin, as I said, is used for a lot of superficial tumors and is also now used for deeper tumors. But yeah, it's a reality. And in the case of, as I said, of animals, it's very frequently used, or much more frequently than in case of humans, because there is the radiotherapy, in a lot of cases, is not an option. It is a universal phenomenon. It even happens with artificial membranes. If we do an artificial lipid membrane, we are going to observe the electroporation phenomenon.
Yeah, thank you. Well, thank you very much, Tony. Yeah, so I have a few questions. There are curiosities. So when you're doing then the simulations, so are you varying the material properties of the media, and does it affect the interpretation you can have of the phenomena?
I'm not sure to understand what you mean. I mean, what I presented is that basically when now we are simulating electroporation at tissue level, so a refinement that was introduced after some years was to increase the conductivity according to the electric field. So basically what we said is there is electroporation, so this electroporation has to have an impact on the conductivity of the tissue, a local, and this is going to have an impact on the distribution of the electric field. And at the same time, this is going to have an impact on an electroporation, so it's going to have, let's say, a mutual interplay between electroporation and the electric field distribution. And this is something that we are simulating now, and what I showed with the potato, it indicates that, yeah, this is true, and it's working. And basically it was motivated, this study was motivated because when we were doing the simple assumption that the tissue conductivity was constant, in some specific electrode setups, we saw that there was a disparity between, quite significant disparity in animal studies between what we expected and what we saw. And so we understood that there was something missing there, and we know that now we know that it's this change in the conductivity that we have. This conductivity change has to be modeled carefully, of course, because it's an interactive simulation and it can reach instabilities. But now we know how to do that.
Okay, so basically then depending on whether there are tissues in the human body that are not electrically neutrals, and then they require the diffusion of mobile ions to become electrically neutral. So if you have then an electrical field applied to it, so it changes a lot of things. And plus you have a lot of collagenous molecules, depending on the configuration, that becomes piezoelectric. So that would be true for bone, that would be true for cartilaginous tissues, proteoglycan-rich tissues. So can it be something important to take into account, or is it simply too much of a detail and you don't need that information?
Yeah, that's interesting. As far as I know, nobody introduced the piezoelectricity possibility in the case of bone. It's true that electroporation of bones has been done, and it's useful for treating metastases in the bones. But as far as I know, piezoelectricity has not been modeled in the context of electroporation. We basically, in all our models, assume that tissues are ionic conductors. Okay, so basically, either we assume that they are pure conductors, or they have some dielectric properties. In particular, when we model at cell level or at micro level, there we say, okay, we have the membrane, which is a dielectric, more or less perfect, and then we have the intracellular medium and the extracellular medium, and we assume that the intracellular medium and the extracellular medium are conductive. We always assume that they are conductive and they are neutral. We always assume that. I don't know if anybody went a little bit deeper in this sense. I mean, at molecular dynamic simulation level, yes, of course, because there you have to add the ions to have conductivity, if otherwise. But,
okay. So it's more a question of basically the electrical properties of interstitial fluids that matters. So, and then, Bart, you have a question. In the meanwhile, that's another question is, so if you transfer then these new methods, for example, the H-fire, and you want it to be adopted as clinical protocol, so would you use digital evidences in order to back up the efficiency, the safety, etc.?
Well, there is an interesting thing that around that has nothing or not much to do with science. I mean, I don't know if I can go back. The people that is doing this PFA, which is, I called you the pulse field ablation for cardiac ablation. They call it PFA, and they don't call it irreversible electroporation because they wanted to avoid or try to avoid issues with patents. And the second thing that they do is that they don't mention the protocols at all. So which is the waveform that they use? I mean, they are publishing in journals, and this is something that a lot of us is really getting us mad. They publish in very prestigious journals without specifying which is the shape or the waveform that they are applying, the electric field waveform. And it's very important that. And what we know now is that they are using H-fire. But they, of course, they don't want to say it because there is a patent on H-fire. We can discuss if it's more strong or less strong or more strong, that patent. But in order to avoid any issue, or that's how I see that, or one of the reasons I see it, they don't publish that. So they are already using H-fires because otherwise PFA, I mean, the use of electroporation for cardiac applications would not make too much sense. I mean, if you want the ablation until now, it's basically with radiofrequency ablation. It's performed with sedated patients in a lot of cases, not all, but in a lot of cases it's performed not with anesthesia, with general anesthesia. So by using H-fire, you can use the same clinical setups, basically. You don't have to do big changes to the common practice. So H-fire is already being used, although it is not explicitly mentioned. So there is no doubt that it's already used by companies.
Thank you. Very, very interesting overview. It's like when you do the simulations, you assume or you kind of very simplify the tissue in some ways.
Correct.
So my question is a little bit, when you look, for example, at vasculature, presence of blood, of flow, or damage in microvascular that you would induce, how important is this?
A very good question. That's important. That's important. Well, basically, have not, but we in the past performed some simulation more at, let's say, a small scale. So people has done at different scales, cell scale, let's say microvascular scales. There, yeah, you have to include the different properties of the smooth muscle cells and the intima and everything. It's important. It's important in particular because when we do these simulations, one thing that the simulations reveal is that the endothelial cells for sure are damaged, okay? And it's true. I mean, we know that when we do electroporation of blood vessels, the endothelial cells are gone. And this is something that a lot of people would say, wow, this is super dangerous, you cannot do that. The nice thing about electroporation, and including irreversible electroporation, is that yes, we remove the, as I said, this experiment was, this observation on the right was done 1 week later. But after this study, another one was performed they studied what happens 1 day, 3 days, 5 days, 1 week after. And what they saw is that when we apply electroporation, the smooth muscle cells are gone. They are not there anymore. What happens is that because all this extracellular matrix is preserved, they migrate, they repopulate very, very rapidly. So that's something that we know. And this is, as you suggested, this is anticipated by the simulations. Another thing that we know is that because we damage the endothelial cells, there is a collapse of the small vasculature. So the small vasculature, in a lot of cases, disappear, red blood cells go everywhere, and it collapses. So in that region, there is ischemia. And we believe, and in particular in my case, that in a lot of cases when we treat tumors by IRE, in the particular case of the liver, in most of the cases, we have this additional benefit of ischemia, or even it's the major driver of killing the cells. Yeah? So that's a very good point.
Okay, thank you very much.
Well, let's take one last question.
Okay.
I have a question with regards to the fact that for the treatment against cancer, the fact that the extracellular matrix that the cancer cells might generate, if this is not destroyed, is it meaningful or it doesn't matter as much?
Well, we want to be sure that we kill the tumor cells. What we want is not to leave, for instance, a hole. I mean, we don't want to create fistulas. And the nice thing about irreversible electroporation is that, or electrochemotherapy, is that we kill the tumor cells, and if the healthy cells basically should grow again, or it happens, they grow again and repopulate the area that has been treated. For instance, in the case of nerves, I said, it's very commonly used for prostate, or quite commonly used for prostate, because it does not damage nerves. We know that nerves are damaged, and actually, there is some, I mean, the function of the nerve is disrupted for hours or even days. The nice thing is that because we have myelin, so what is the extracellular matrix of the neuron is completely preserved. What happens is that the axon regrows again very rapidly across this myelin, or what it seems to be happening. So we preserve the extracellular matrix. The cellular matrix is not going, in principle, to promote the growth of tumor cells. What it's going to promote is the growth of healthy cells if we destroy all the tumor cells, of course. It seems, of course, there is a lot of discussion about how important in all these treatments is the immunoreaction, of course, and there is a lot of hope thinking that because we destroy the cells, we release the intracellular contents, this is going to promote the immune response against tumors, and people has been working on further promoting the immune response, but there is still a lot of unknowns around that.
Okay, thank you.
Thank you.
Thank you very much. Unfortunately, we don't have any time for another question. So thank you for the talk again.
Thank you very much.